People are claiming Epitalon can massively extend human lifespan, but could its anti-aging mechanism actually create a hidden cancer risk? Your body does not age through one mechanism. I've actually discovered 12 mechanisms of aging, which I show in the video linked in the description. Some of them include your cells losing access to the right genetic information, your cells gradually losing their ability to divide, your immune system becoming less effective at identifying damaged or abnormal cells, mitochondrial dysfunction, and the biological clock that controls sleep, hormones, and metabolism becoming less precise. Epitalon is unusual because it interacts with several of these systems. It has been shown to improve telomere length, restore nighttime melatonin production, and affect several systems involved in cellular regulation. But its effect on telomeres is exactly where the concern begins. Telomeres protect the ends of your chromosomes and help preserve genetic information inside your cells as they divide. Each time many of your cells divide, those telomeres gradually become shorter, and once they become critically short, the cell can lose its ability to continue dividing normally or even lose access to some of the information. That matters because your body is constantly renewing itself. Many of the cells that you have today will eventually be replaced by new cells throughout this process of cellular division. But cancer cells face the same limitation. In order to keep dividing indefinitely, they also need a way to maintain their telomeres. So to understand whether Epitalon's effect on telomeres is protective, risky, or more complicated than either of those labels, we need to understand what Epitalon actually is and why aging is about much more than just telomeres. Most people hear one thing about Epitalon, telomerase. It activates it. And immediately that raises a concern because telomerase is also active inside cancer cells. So the assumption becomes, wouldn't this make cancer worse? That's the wrong way to think about it. Cancer is not just uncontrolled growth, it's loss of regulation. Cells stop responding to the systems that normally control them. Apoptosis, immune detection, and gene expression balance all begins to break down. Epitalon doesn't just activate telomerase. It affects gene expression at a higher level. It induces deheterochromatization, which reactivates regions of DNA that have become locked down through the aging process itself, or in the case of some diseases. And all of this has much more of a broad effect on biology. Telomere maintenance itself is not inherently good or bad. In a normal aging cell, restoring telomere maintenance can preserve its ability to keep dividing. In a cancer cell, that same general capability can help the cell survive and continue replicating. And you see this kind of context dependence with other regulatory proteins as well. Thymosin beta-10, which is induced in some instances in the lab, has been associated with cancer cell apoptosis in some of the models, yet progression in others. But even Epitalon doesn't seem to affect every cell the same way. In normal human cells, it significantly increased telomerase activity. In 2 breast cancer cell lines, telomerase activity did not significantly increase at all. Instead, those cancer cells relied more heavily on a different telomere maintenance pathway called ALT. And you see this same context dependence elsewhere throughout biology. One example is GDF11, a protein that has shown tumor suppressive effects in several cancer models while producing very different effects in others. That's important because biology doesn't classify a pathway as simply good or bad. Telomerase isn't inherently cancer-causing, GDF11 isn't inherently anti-cancer, and thymosin beta-10 isn't inherently either one. What matters is the cell it's acting in, what other regulatory systems are active, and what the cell is actually being instructed to do. To put this into perspective, exercise also induces telomerase downstream through irisin, which is released by muscles during exercise. Exercise also increases other pathways associated with cancer growth, such as vascular endothelial growth factor. You'd think that exercise would then be bad for cancer. Yet it's generally considered anti-cancer, and when researchers took the blood of those who exercise regularly, it was shown to suppress certain types of cancer growth, whereas the control's blood did not. This is why in multiple animal models you see something that might seem contradictory at first: reduced tumor development despite telomere maintenance. Because the outcome is not driven by a single pathway, it's driven by whether the system as a whole is functioning correctly. And there are some pretty striking numbers behind that as well. For instance, in mice that were genetically predisposed to specific types of breast cancer, Epitalon reduced the maximum size of the breast adenocarcinomas while also reducing the specific gene expression inside the tumors that control progression. And in a chemically induced colon cancer model, the number of colon tumors also decreased with Epitalon versus the control groups. And another long-term mouse experiment gives us an important bit of nuance here. Epitalon did not significantly reduce the overall spontaneous tumor incidence, but leukemia development was sixfold lower, while chromosome abnormalities in the bone marrow cells were reduced by 17%. So the evidence isn't simply that telomerase goes up and cancer goes up with it. The biology appears to be much more complicated than that. And this is where most people go wrong. They hear something like this and then they try to reduce it to a yes or a no question. Is this good for cancer or is it bad for cancer? But that's not how this works, because loss of cellular control can come from completely different places. Immune surveillance failure, accumulated DNA damage over time, environmental and toxic exposure, and even your genetic susceptibility, and so on. And this is exactly where you need to consider the precision cell method, because instead of asking, what does this compound do? You start asking, what is actually breaking regulation in my system? Or this system? Because Epitalon is targeting one layer, which is the regulation layer. But that's not the only layer that determines an outcome. Consider its effects on the immune system. Epitalon improves immune function, including T lymphocyte populations, which are directly responsible for identifying and eliminating abnormal cells. So now you have two things happening at the same time: improved cellular regulation and improved immune system surveillance. And that combination is far more important than simply killing cancer cells. It's preventing the environment that allows them to take over. There's also the pineal gland component to this, because Epitalon restores melatonin production, and melatonin is also deeply involved in antioxidant defense, DNA protection, and immune modulation. So when that signal is restored, you're reinforcing multiple layers of protection simultaneously. Now, to address the concern directly, there are studies showing that telomere maintenance can increase certain cancer cell lines with Epitalon. Now, to address the concern directly, there are some studies showing that telomere maintenance can increase in certain cell lines with Epitalon. That's real, but that's isolated cell line behavior, and it's not the same as whole system regulation in a living organism. And the broader body of evidence shows something very different: improved regulation, not runaway cancer growth. And this has been seen in multiple different animal models with Epitalon. There's another interesting finding with Epitalon as well. It has been shown in the lab to increase the number of mesenchymal stem cells by helping the cells continue to proliferate and reducing markers of cellular senescence. Basically, it helped keep the cells from acting as old. This is another example of Epitalon affecting the regulatory machinery of aging and potentially helping to support tissue repair all throughout the body. Now, in regards to dosing, 100 micrograms is the start. Epitalon has been shown in primate studies to increase nighttime melatonin production, and it did that at 100 micrograms. So what I recommend for people in general is 100 micrograms to 600 micrograms, either through injection or intranasal administration. I don't recommend above that because, in our experience, it can induce temporary agitation in some people. Now, you may hear some people recommending to take up to 10 milligrams of Epitalon for a short course, but that is not data that comes from the literature. That is data that comes from Epithalamin, which is the full pineal gland peptide complex. It has been dosed at 10 milligrams in the literature, but it was also shown that just 100 micrograms of Epitalon was as potent as 50 milligrams of Epithalamin at inducing melatonin production in primates. So people who are working with me on a more personal level typically get the same recommendation, 100 to 600 micrograms. And if you want to learn more about the pineal gland and pineal gland peptides, you can get our book, Peptide Salvation. The link is in the description below. And my name is Brendan Henry. I'm one of the founders here at the Peptide Science Institute. I want to thank you so much for watching. Please do leave a comment below. I read all of them, even if I don't always reply. And I hope you have a great day or night.